Welcome to all my friends who are joining us on the recording. Today is March 25th. We are about to get started with our session today. As we're getting ready to get started, I'm going to do a couple of quick polls here in the classroom like I always start with, just to give me some ideas. So my friends here joining me in class today, I am posting up a question for you. And give me a minute, I'm going to give you a poll. I want to know if today is your first time in office hours or if you've been here before. You should see a little thing pop up on your screen. Just click for me A if it's your first time or B if it is not your first time. Oh, awesome. We are getting great responses here. Okay, so most of us have answered in the poll. I'll show you guys. Most of us have been to office hours before, so that's great. Welcome to all my new friends. And again, welcome to my friends that are here on the recording. So looks like over half of us have been to office hours before. Some of us are new. This will be perfect. So I'm going to end that one. As always, I'm going to ask again when our lab was before the switch to online. So I'll give you a second to read it. And here comes that poll. Before the world went crazy, when did I used to see you for lecture? For a while there, my poll was hanging out halfway in the middle. Nobody was answering. It's like, yeah, we're to the point where half of us have forgotten when we used to have lecture, right? It's been so long. It's been a long time. All right, so I will show you guys. We are mostly friends from Monday and Wednesday. Since it's Wednesday, I'm not surprised. That makes a lot of sense. What I always say in office hours is if I say something today during office hours that you think would be really important to let your classmates know about fast before they sit down and watch the recording, please send that out in your class group me just to make sure that everyone gets that information fast. So my one friend from Tuesday, Thursday, you're on the hook for helping out your friends in that section. But if there's anything really important that you feel like you should pass along, please let them know in group me what we talk about today. One more question. And this question will help me out here to know where we are at in our preparations for today. I want to know if you have worked on all of lesson number 9, some parts of lesson number 9, or if you haven't started it yet. And by the way, this is completely anonymous. So if you haven't started yet, I'm going to have no idea it's you. You're totally fine. So let's see where we are at right now. Okay, I think my votes are in. I'll show you guys. A little over half of us have done at least half of lesson number 9. Some of us, that's perfect. What I am going to share with you now, as opposed to these questions, is I want to show you something that you have access to, especially for my friends that this is your first time in office hours. Each week, I'm going to post for you a schedule of what we're going to be doing in office hours each day. So today in office hours, primarily what we're going to focus on is on Wednesday, March 25th. Most of what we're going to talk about today is stuff from the second half of lesson number 9. So in lieu of doing lectures like we would do in class together, what I've built for you guys is step-by-step activities that go through the information just like we would have gone through it during lecture class. So when you see on the schedule for next week, when next week's schedule comes out, when it says these page numbers, that refers to the pages inside the lesson. So this week's lesson, lesson number 9, the muscular system, that had 22 pages. That's where this number 22 came from. So these page numbers are specific pages inside the lessons. For today, I'm primarily going to focus on the content that was in those second half of the lesson. But if we have time and if there are questions about some of the stuff from the first half of that lesson, that's primarily what we covered yesterday. If you couldn't make it to office hours yesterday, I would definitely recommend watching that video. We did some good stuff with what's called excitation, contraction, coupling, kind of the big picture, almost everything that you were doing in the first half of the lesson. So the recording from yesterday's office hours focuses on that. We're going to focus on the information from the second half of that lesson. As a heads up, tomorrow is going to be the lab day, the main lab day for me this week. So what you can do to get ready for tomorrow's section is to start working in lab activity. That's going to be very similar to the lesson number 9 that you're doing for lecture. So the week 11 lab activity goes through page by page, exercise by exercise in your lab packets to help you complete those activities like we do in class. So please work on that when you come to our office hours tomorrow. Then we'll be talking about that. Kylie asked for some of my Friday lab friends because they have Dr. Adams. You are welcome to come to either of the sessions. She will probably do very similar or maybe not, I don't know, to what I'm doing. So no pressure to attend. If you want double coverage, feel free, but you're never required to attend office hours. So if you don't want to attend the lab ones, there's no hard feelings. I totally get it. So for tomorrow's office hours, same time, same place. Yep, I'm always recording our office hours. So tomorrow, some lab work, bring that lab packet with you. We'll do some lab work during our office hours tomorrow. And then Friday is what we see here is just going to kind of be a general wrap up session. So I will take lecture questions. I will take lab questions. It'll kind of just be like a popcorn. What is it that we want to hash out together kind of thing on Friday. I had a question about the numbering of the lab that we're on. It is on your packet going to say week 10. We're calling it week 11 because that's what week we are now. So just imagine that the number at the top of the packet is wrong. That's the only thing that's changed. All the content is exactly the same. So when you go into the Google Drive now, it's going to say that it's week 11. When you open it up, it's going to still say that it's week 10 because that's what it was uploaded as. So week 10, formerly now called week 11. That's what we're doing in lab this week. You're welcome, Leslie. All right, so for today's office hours, we're going to focus on lecture. Tomorrow we'll do lab. Friday is going to be a mix of whatever we want it to be on Friday. Let me pull up really fast, since some of us are here for the first time. You have an updated lecture calendar posted for Lecture Resources. To give you an idea of where we're at right now, we are right here in week 11. So the thing that you must get done this week, as in it's due by Sunday night at 11:59, is the lesson number 9 homework. That must be done. So all of our studying with the muscular system, the way that muscles contract, the different ways that I power muscle contraction, I saw someone mention that, that kind of stuff, all due by Sunday night at midnight. I am in the process of wrapping up the second lesson, neuron physiology, that's going to post for you by end of day today. So you will see that next lesson coming available. I will start doing office hours about that starting next week, and I will get you an updated schedule about neuron physiology. Today is Wednesday. It's hard to keep track, right? So neuron physiology is going to be your primary focus next week. I will also be getting posted for you about midweek next week our last lesson in unit number 3, and that's going to be about the brain. So 3 lessons that will be on exam number 3. That's going to be this muscular system lesson, the neuron physiology lesson, and then the brain lesson that will pop up next week for you. If you had not already heard all lab assignments for all sections, this is my Monday friends through Friday friends, and this is Dr. Adams' friends too. your lab assignment for each week is always now Monday at 11:59. So if you were a Friday lab friend and you didn't have to do lab homework until Thursday night, we need to make a mental shift in our mind that lab homework is now due on Sunday night, which is why I'm going to host a session on Thursday, and probably next week I'm going to host a session a little bit earlier to do our lab review stuff. So please don't miss out on week 11 homework for every single student in every single section. Do Sunday night at 11:59. So new timing on that. I had a question on the date of the exam. We talked about this on Monday. It's going to be sometime in the window of April 8th through the 10th. I will get you some more details about that. What I think is the way it's going to work is it's going to be open for 24 hours. And it's going to be open on whatever day of the week your normal lab day was, or your normal lecture day was in that time, excuse me. So for my friends that are Monday, Wednesday class, you'd be taking the exam on the 8th. For my Tuesday, Thursday friends, yours would be open on the 9th. And for my Friday friends, it'll be open on the 10th. So that's the tentative plan. But I will let you know as I have more details about that for sure. So sometime in that window, April 8th through the 10th, that's going to be when the first lecture exam is online, exam number 3. All right, I need to go back to my chat and see what topics. I didn't get a lot of topics from you guys. See what we're thinking. I see a question in here about the homework. Yes, so the homework, we remember for lecture homework, you now have unlimited attempts on the lecture homework. So feel free to do the lecture homework. If you don't get full credit, if you need to go back and try it again, feel free to do it as many times as you'd like. So yes, you can go back to the modules, review some more information, review the recording of the office hours, go back and fix that. So that will be available for you. Let's see, Kylie, you don't have to be alone in a room to take the exam. I feel you that you have other people in the house and it's not empty. So you don't have to be alone in a room to take the exam. That is correct. And the modules are going to stay up forever. So the modules will be up as you are, as you're reviewing for the final exam. They will still be available for you. So the modules are not going anywhere. I lost it in the chat. Help me out here. A couple of you had told me some topics you wanted me to talk about. I know one of you talked about power contraction. So my 3 types of muscle tissue, muscle contraction. I know I had a friend yesterday who wanted to talk about acetylcholinesterase. I can't remember who that was. If you're here from yesterday and that was you, can you either wave at me or say that was me in the chat box? Somebody want to talk about that. Perfect. Okay. So today, Isabella, we will have time to talk about acetylcholinesterase. Any other topics that we need to work on? Sounds great. Yeah. Okay. Well, what I'm going to do then is I'm going to start with the end of the packet. I'm going to start with the stuff about powering muscle contraction, and then we're going to swing back into acetylcholinesterase, some of those disorders that we're talking about. Let me just see for my reference. I'm going to create a little poll, make it anonymous for us here. Here is the question. I have not worked on the second half of lesson number 9. True or false? This is completely anonymous. I just need to know where we're at. I need to know how many of us haven't had a chance to cover this material at all, because that's gonna make our office hours a little bit harder today. Yes, lesson number 9, it is the lecture number 9 packet. When I say the second half of it, I'm meaning those pages in the lesson online that are pages 12 through 22. Yeah, so if you finished everything, then your answer to the question would be false. You've done everything. Okay, so I'll show you on my side what I'm seeing here. And again, it's anonymous, so I don't know who's who. We're about 50-50 split. So for my friends who haven't done the second half of the lesson yet, please, after office hours today, start trying to work through that. Some of the stuff I'm going to talk about today might not make a ton of sense to you since you haven't had a chance to look over the things before. Best to help you out. Yeah, with the critical thinking questions. So I'll try to give you some guidance. But if you guys remember way back to when we used to do class, I didn't always give you critical thinking question answers either. So I'll try to give you some scaffolding. I'll try to help you out. So we'll see if maybe we can work together as a class to get to some of those critical thinking question answers. So let me go back to my PowerPoint. Actually, I probably should just do a whiteboard. Yeah, so I'll show you a quick picture just so that I have easy access to it later. And I'm going to pop up a whiteboard. So when you think about the types of skeletal muscle tissue in the body, we have 3 types of skeletal muscle tissue. So we're going to do whiteboard here together. So skeletal muscle tissue is what we're talking about today. That would actually be something for you to make a note of for yourself. At the end of lesson number 9, or it's the lecture number 9 packet, the 3 types of tissue, so slow oxidative, fast glycolytic, those types of tissue are only seen in skeletal muscle tissue. So this is something that's very particular to skeletal muscle tissue. We have 3 types of skeletal muscle tissue. So I rattled them off really fast. Now I'm going to go a little bit slower for you here. So one of the types that we have in the slow oxidative muscle fibers. Hey, here's a question that I know that we can answer because this was lab a couple weeks back. When I use the word muscle fibers, what's the normal kind of tissue word for this fibers thing here? A muscle fiber is a muscle, yeah, Leslie said, yep, mention it for us. It's a muscle cell. Okay, so the first kind of muscle cells that I have in skeletal muscle tissue, I call them slow oxidative muscle cells or muscle fibers. The next kind of muscle cells or skeletal muscle fibers that I have are called fast oxidative muscle fibers or muscle cells. And the final kind of skeletal muscle fibers that I have in the body are called fast glycolytic muscle fibers. Fast glycolytic. So all of the skeletal muscle tissue falls into one of these 3 groups. We can change from one group to another. That's something for your kinesiology teacher to talk to you about. We're not going to go into that. But what we want to know for the sake of our class with these types of muscle tissue is 2 main things. We want to know first how quickly they contract. The reason we want to know how quickly they contract is that tells us how strong their contraction is. So the first thing we know is this something that's going to contract very quickly or something that's going to contract more slowly? In general, for my types of tissue that contract quickly, those are going to be stronger muscle contractions. My muscles that contract more slowly, those are going to be less strong muscle contractions. So the first thing we want to know about our skeletal muscle tissues is how quickly do they contract. The second thing that we want to know about our muscle tissues is how they, wow, this is just terrible, how they make ATP. I know we can answer this question because we talked about this in lab and in various things. When I talk about ATP, what is the purpose of ATP in any kind of cell? Not just a muscle cell, in any kind of cell. Why do I need ATP? Absolutely. Yep, we're all popping up. ATP is energy. It equals energy. For you to do the process of muscle contraction, it's going to take energy. That should be no surprise, right? We're saying how we miss our gyms, right? But being at the gym is something that took a lot of energy, right? You were using energy to exercise. So when we talk about the types of skeletal muscle tissue we have in our, there are two things that we want to know about it. The first thing we want to know about it is how quickly it's going to contract, or to put it another way, how quickly it can chop up and use ATP. If you didn't get a chance to attend office hours yesterday or to watch our video about office hours, we took some time at the very end of office hours to talk about energy of ATP. So highly recommend that for you. If you don't, if you were in one of the classes where we didn't get to talk about the energy of ATP in class, make sure to take a look there at the end of office hours from yesterday. Okay, so when we're talking about our 3 types of skeletal muscle tissue in the body, how quickly they contract and how they make their ATP, the good news is the names of these types of skeletal muscle tissues tell me all of that information. I don't have to come up with it on my own, and really I don't have to memorize it even, because the name of muscle fiber tells me this information. So when I'm talking about my types of muscle tissue, when I want to know how quickly they contract, it's not a trick question. It's the first part of their name. So not a trick question here. My first type of skeletal muscle tissue is called slow oxidative skeletal muscle tissue. When I talk about slow tissue, am I going to contract quickly or slowly? I promise it's not a trick question. Yes, exactly. A few of us are chiming in. It's one of those questions that if I was in class, I bet that some of you would have been like, is this a trick question? I promise it's not. So when I talk about slow oxidative muscle tissue, it contracts more slowly. When I use this word slow, what I'm actually referring to is the activity of an enzyme called ATPase. ATPase. I know it's been a while since we talked about enzymes and proteins, but if you remember from when we were talking about enzymes and proteins, usually their name tells me exactly what they do. When I talk about ATPase, that ending part ACE tells me it's an enzyme, and the beginning part tells me what it's working on or what it's chopping up in this case. So the enzyme ATPase, its one job in life, is to break down ATP. Absolutely, Leslie. That's what I was literally going to type. ATPase. It's an enzyme that breaks down ATP. When I break down ATP, so break down ATP, that's going to give me energy. That's the reason that we build ATP in the body, because when I build it, then I can break it and it releases energy for me. So the first part of this type of muscle tissue, slow oxidative muscle tissue, that tells me that this particular type of muscle tissue, its ATPase, its enzyme that chops up ATP, works a little bit slowly. So I've got 2 other types of muscle tissue over here. Question, how quickly are these other 2 types of muscle tissue? How quickly is their ATPase going to work based on their name? Exactly. Yep, we're chiming in fast. It's going to work quickly for me. Absolutely. So when I talk about my fast oxidative muscle fibers, or when I talk about my fast glycolytic muscle fibers, the fast part of their name just tells me how their ATPase works. So remember, here's some big ideas for us. Remember that this first part of their name is telling me how quickly that muscle cell can contract. If a muscle cell can contract quickly, that's going to mean that it generates a strong contraction. Because remember, the other thing we said is the strength of the contraction comes from how quickly it happens. If I do a really fast muscle contraction, that's going to be a really strong contraction. If I do a slower contraction, that kind of contraction is not going to have as much energy. The word fast or slow specifically relates to this ATPase that we talked about. That's what I use to break down my ATP. So the first thing we need to know about all of our types of skeletal muscle tissue: how quickly do they contract? Are they fast? Are they slow? Again, you don't have to memorize that because their name literally tells you that. That's perfect. That's helpful for me. The second part of their name tells me the second kind of information I need to know about them. The second kind of information I need to know about them, how they make their ATP. So there are two ways that my skeletal muscle tissue can use to make ATP. The first way that my skeletal muscle tissue can make ATP or make its energy is through a process called glycolysis. Glycolysis. Now, if we were in class, I would take this word glycolysis and I would chop it in half and tell you what the two parts mean. So when I use this word glycolysis, let's start with the glyco part of its name. The glyco part of its name means glucose. Or if we want to get really technical, the glyco part of its name technically means glycogen. And as you're working on lesson number 9, that lesson talks to you about what glycogen is. When you have extra glucose in your body stores it in big, long lines that kind of branch off all over the place. That's what we call glycogen. So think of glycogen kind of as stored energy, or it's stored glucose. So the first part of the name of this process I can use to make energy is glyco. That means sugar, or that means glycogen. The second part of the name of this process is lysis. And this word lysis means we're breaking or breaking down something. So the first way that your skeletal muscle tissue can is by breaking down glucose or breaking down glycogen. Hey, this is not a trick question. We've got our 3 types of skeletal muscle tissue across the top of our screen here. Which of those types that we see up there, which one do you think would generate ATP using glycolysis? Any guesses? Which one might use glycolysis? Yeah, I got 1 vote. Perfect. It's hard to type, right? It's a long name. Yeah. We're all agreeing here. When I talk about using glycolysis to break down or to make my ATP, the type of muscle tissue that's going to use glycolysis is the type that has glycolytic in its name. So glycolytic muscle tissue, we use glycolysis. We break down glucose, or glucose in the form of glycogen that we stored for use later. So that's the first way that I can make my ATP, is using glycolysis. The second way that I can make my ATP is using something called the electron transport chain. And the electron transport chain is something that we actually find inside an organelle in the body. For my friends who already did the second half of me out, which organelle has the electron transport chain inside of it? Does anyone remember? Inside the, see if anyone remembers. I got the crickets, so maybe not. There we go. Couple of us remembered. The electron transport chain is found inside the mitochondria. Inside the mitochondria. Yeah, there we go. That's another long word. To be fair, that was a long word to type. So thank you for those of you that took the time to type it for me. The electron transport chain is inside the mitochondria. If you remember from studying the organelles, we said that the job of the mitochondria was to make energy for the cell. Here's the specific way that it goes through to make that energy. So the electron transport chain is a group of proteins that live inside the mitochondria. Their job is to make ATP, and they're actually really good at making ATP. The electron transport chain makes a lot of ATP. Here's the caveat, though, with the electron transport chain. The electron transport chain, I'm going to abbreviate it here, the ETC must have oxygen. We must have oxygen for the electron transport chain to work. This is because at the very end of the process of making your ATP with the electron transport chain, it's going to give its leftover electrons that don't have energy, it's going to give them away to oxygen. We have to have oxygen present so that at the end of the electron transport chain, I have somewhere to put those electrons that have no energy. So for the electron transport chain to work, I must have oxygen. The main reason I point that out is because that is something that's going to help me to figure out which kind or kinds of skeletal muscle fibers use the electron transport chain. So when we're looking at our types of skeletal muscle tissues here, based on their name, which one or ones do we think might use the electron transport chain to make ATP? What are our guesses on that? Yes. Yeah. So it looks like we're pretty much in agreement here. We're honing in on the fact that it has to do with this word right here, oxidative. Oxidative means that I use oxygen. Yeah, it's okay that you can't spell. I can't spell either. We're all good. Oxidative means that I have to use oxygen to make my ATP. The process of glycolysis doesn't need oxygen. We don't have to have oxygen. But the process of the electron transport chain, we do have to have oxygen for that to be able to work. So big picture, what we've talked about so far, there are 3 types of skeletal muscle tissue in the body. We have slow oxidative skeletal muscle tissue, fast oxidative, and fast glycolytic. For each type of skeletal muscle tissue, the 2 things that you must know are how quickly they contract, or what ends up being how strong their contraction is, as well as the way that they make their ATP. You can tell all of that information from their name, which is excellent. That's helpful for us. Now, I gave you an analogy in the lesson, and it's in your notes packet, to talk about the types of skeletal muscle tissues. And that analogy that I gave you, let me pull it up here from my pictures. The analogy I gave you for these types of muscle tissue is we're going to pretend that our muscle fibers are lumberjacks. So think about our muscle fibers as lumberjacks. Their job is to chop up wood that you can take inside your house and burn in a fireplace or burn in an oven to make food for you. Now, my lumberjack over here on the left, he's a professional lumberjack. He looks more too, right? So this is the guy who knows what he's doing. He's really strong. He does this all the time. Over here on the right, we've got the hobbyist over here who, I mean, it's basically the zombie apocalypse, right? So this guy decided that he wants to ride out this coronavirus thing out in the woods. So he's going to chop some wood, man. He's going to make this happen. He's going to power his house. So he's not nearly as efficient as my lumberjack friend over on the other side of the image. But both of them are chopping up the fuel source, the wood here, to power the house. What I'm going to do is give you a moment. I want you guys to think about what we just talked about related to fast versus slow types of fibers. I want you to try to relate these two lumberjacks that we have in our picture to fast versus slow. And let me show you. Pull up, go to that screen. Bear with me for a minute. If you need a little guidance to answer how my lumberjacks represent this analogy, let me share the specific with you. We are on page 16 of this week's lesson. Page 16 of this week's lesson has our little lumberjack friends here. So I'm going to go on radio silence here for a couple of minutes. I want you to try to summarize in your notes how my two different types of lumberjacks might represent those ATPase enzymes. So pull up page 16 in the lesson, make some notes for yourself. I'm going to give us a couple of minutes, and then I'm going to open up the floor for questions that we have about the speed of ATPase. So I'm going to stop talking here, a couple of minutes. Let's see if we can summarize this. Nora is asking for where you'd find this lesson. When you are, yes, page 16. When you're in the course Blackboard site, click on lecture resources, and it says, let me pull that up and I can help you out. So when you're in the lecture resources folder, there's a folder that says online learning modules. Inside that folder says lesson number 9. Resources, online learning modules, lesson number 9. That's where we're at. Yeah, thank you, Eileen. It looks like in your lecture packet notes, it's on page 11. We are on where that 16 came from is at the top of, if you notice on the left side of the screen, I'm not sure if you can see my mouse hovering, but on the left side of the lesson screen, it says page 16. But yeah, it sounds like that picture is on page 11 of your lecture outline. Yeah, so let's get some questions in there. Kylie's got a good question that I'm going to address. Let me give you a minute more here. If you have a specific question right now about fast versus slow, go ahead and type that in. I'll answer Kylie's question while you're typing. Kylie asked, both fast and slow ATPases lead to muscle contraction, correct? Yes, that is correct. Think about our two lumberjacks. It doesn't matter if you chop the wood really fast or if you chop the wood really slow, you're still chopping the wood. We can still bring that wood into the house. So yes, Even if I have a slow ATPase, it will still help me to do muscle contraction. That is correct. Yeah, fast just does it more quickly. Absolutely. Fast takes that energy that's in the ATP, it chops it up, and when I chop up the ATP, that frees up the energy for me, and I can use that energy to do a contraction. Yeah, so Alexis is asking if we do use a fast ATPase, does that mean we have a stronger contraction? That is correct. Yes. A fast ATPase will have a stronger contraction. Absolutely. Yeah, so Carrie is asking a question that pulls us into a little bit lower on that page. Let me scroll down a little bit farther. What we haven't talked about yet related to fast versus slow is how quickly you would run out of your ATP using these two kinds of enzymes. So no, totally fine, Carrie. This is where we're going next. So you're good. When we talk about how quickly I can chop up ATP, you do also have to consider that for some types of muscle tissue, I don't have a lot of ATP. So if you're really good at chopping, and you've got, let me scroll up a little bit more here. If you're really good at chopping, like my friend over here on the left, the strong one, he's really good at chopping wood. If you give him a wood pile with 3 logs, he's done in like 3 seconds, right? He's just going to chop those logs in like 3 hits and he's done. My hobbyist friend on the right, it's going to take him a little bit longer to chop up that ATP. So one of the things you have to consider, we didn't talk about it a lot when we were on our previous whiteboard with fast versus slow and that kind of stuff, is if you have a fast ATPase, you will generate or liberate ATP really fast. But what's not going to happen, or what is going to happen, excuse me, is if I have a limited amount of ATP, it's gone really quickly. So a fast ATPase burns through the ATP more quickly. I like that terminology because we're talking about firewood, right? If I chop it up really fast with a fast ATPase, my supply goes away really quickly. Actually, that's really a great analogy for right now, right? So where we bought food, hopefully, to ride out the apocalypse. And then I brought my mom and my high school senior brother are staying with me right now. We got some fast ATPase action going on over here with how quickly we're going through our food. So yeah, think about a fast ATPase, you're going through things more quickly. A slow ATPase is going to go through a little bit more slowly, your supply of ATP. Would this be the same thing that happens during labor? So the challenge with comparing it to labor is that involves smooth muscle. However, how we power smooth muscle is the same as how we power skeletal muscle. So yes, we are chopping up ATP, any kind of movement, whether it's smooth muscle, skeletal muscle, cardiac muscle, we're always using an ATPase to chop up ATP to make energy. So in that regard, yes, ATPases are the same across the board. Their job is always to chop up ATP. Okay, so hopefully, let me go back to my share screen really fast. Hopefully you have answered that question on page 11 of your notes packet about fast versus slow with our lumberjack friends. Pulling back up for you over here with our lumberjack friends. The other thing, so here, let me back up and I'll give you some time to type because this is a longer question here. When I talk about the lumberjacks, we said that these were representing what about my muscle tissue? The analogy of the lumberjacks, what kind of information does this example show me about muscle tissues? Yeah, that's right, Kylie. I did a bad job asking the question and you figured me out. What my lumberjacks are representing is the ATPase. And remember, I told you there's 2 things we got to know about skeletal muscle tissue. We need to know how quickly it contracts, or in other words, how quickly it breaks down ATP. The kind of lumberjack that you have. Do you have the professional lumberjack, like we see on the left, or do you have the hobbyist lumberjack, like we see on the right? That's fast versus slow. The first thing you need to know about your muscle tissue, how quickly does it chop up ATP? That's the first thing. The second thing that I told you needed to know about our types of skeletal muscle tissue is how they built their ATP. So this is a little bit lower on that page for you. Lower down on that page, you have a picture of our little pile of wood, like we were looking at between our two lumberjacks there, versus a forest, like we see over here on the right side of the picture. We didn't talk a lot about the electron transport chain related to how much ATP it makes. So let's make some notes for ourselves about the electron transport chain and about glycolysis. Let's start with the electron transport chain. Remind me, when I told you about the electron transport chain, did I say it made a lot or a little ATP? Can anyone remember? Does the electron transport chain make a lot or a little? Yes, yeah, we're agreeing. The electron transport chain makes a lot of ATP, absolutely. So I'm going to abbreviate here. The electron transport chain, ETC, makes a lot of ATP. In some regards, it's almost an unlimited amount of ATP. As long as the electron transport chain is running, it makes a ton of ATP. Numbers that I won't ask you about on the exam, but I'll tell you just for your reference. When the electron transport chain goes through one cycle, where it does what it's supposed to do once, that's going to generate 30 to 32 molecules of ATP. 30 to 32 molecules of ATP. Now, it's hard for that number to sound like a lot until we go over here and we compare it to glycolysis. In the process of glycolysis only ends up making a total of 2 ATP. Makes about 2 to 4. We'll give it a little wiggle room. 2 to 4 ATP, molecules of ATP. Now those numbers look more impressive, right? We don't need to know these numbers. Yeah. That's awesome, Kylie. Electron transport chain, or train. Yep. We don't need to know these numbers. We do need to know that the electron transport chain makes a ton of ATP. Glycolysis doesn't make nearly as much ATP. So glycolysis only makes 2 to 4, a very small amount of ATP. That's still energy that the cell can use. It's not a lot of it, but it's still energy that the cell can use. The electron transport chain makes a lot more of energy. But there's a couple of caveats with the electron transport chain. The first thing that makes the electron transport chain that we have to keep in mind when we talk about the electron transport chain, I told you guys the electron transport chain needs something. What did I tell you that the electron transport chain cannot work without? What does it have to have? Exactly. Yep. ETC, electron transport chain, needs oxygen. It cannot run without oxygen. So if we're not getting oxygen to our muscle tissue, the electron transport chain cannot make all that ATP for me. I got to have oxygen to be able to do the electron transport chain. See, glycolysis does not need oxygen, which means that glycolysis can basically always happen. There's nothing, no kind of prohibitive factor that says it can't be happening. So glycolysis doesn't need oxygen. The electron transport chain does. The electron transport chain, when there's oxygen, it goes nuts and it makes me all kinds of ATP. But when I don't have oxygen, it's worthless to me. Now, the other thing with the electron transport chain is I guess I haven't run here, so I'll write on here, the electron transport chain makes ATP, emphasis with the makes on it, time. It takes time. We are building something from scratch. We are using, literally, we're taking little electrons and putting them from one protein to another protein to another protein. It just kind of goes down this line of proteins, and we're building completely from scratch, out of nothing, ATP molecules. So it takes me a little bit of time, or there's a little bit of investment involved in getting the electron transport chain started. Once I get it started, once it starts making ATP, it's going to be able to make ATP for me for forever, as long as I have oxygen. The thing with glycolysis is glycolysis uses stored energy. Glycolysis uses stored energy. Now, what I mean by that is what I break down in glycolysis is already in the body. Let's get my spelling right. When I do glycolysis, what did we say we were breaking down in glycolysis? Yeah, there we go. Glycogen or glucose. It's the same thing. So glycogen is a bunch of glucose put together. So glycolysis uses stored energy. That stored energy, I technically store it as glycogen, but it is glucose. It's a bunch of glucose collected to each other. So the thing with glycolysis is I've already got the energy present in the cell. I'm storing it. It's right there. I just need to chop it up, and it's gonna liberate that ATP for me. With the electron transport chain, it takes me a little bit more time since I have to start building the ATP from scratch. It'll take me a little bit longer, but once I get going, man, I get going, and I build all kinds of ATP. So in that regard, I can do contractions a little bit faster if the way they're building their energy is because of stored energy that's already there. If I'm using glycolysis, I've already got that energy present. I just have to chop it up, basically, is what glycolysis does. But here's the thing with glycolysis. Glycolysis is going to be limited in the amount that it can make because we have to start with stored energy. So if I run out, I'll type this for us here. If I run out of glycogen, if you're living at my house and I now have 4 adults here instead of just 2, man, my toilet paper is going faster, right? So if I run out of glycogen, there's no more energy. There's no more ATP. As soon as that stockpile is empty, we're done. So glycolysis can generate energy for me really fast because I'm chopping up energy that was already there. But when I run out of that energy, when I've chopped through, like our picture shows here, when I've chopped through those 3 logs, that's all I had. I don't have more that I can go chop up. Compare that to the electron transport chain over here, what we're representing as the forest. The forest has a ton of wood inside of it. You're going to have to work a little bit harder to chop up that wood to bring into the house. But once you chop down that first tree and start chopping it into small pieces, you've got energy to take into the house as well. So we have almost an unlimited supply of ATP when we talk about the electron transport chain, or an unlimited energy source, versus with glycolysis, we are limited to just what it is that we already stored inside the cell. I saw a question pop up that I lost. Let me go look at it. So is it safe that a glycolysis-powered contraction is a fast, strong, but short-lived contraction, since there's not a lot of it? Yes, I like the way you phrased that, Kylie. So Kylie said if we use glycolysis as our way to liberate energy, number 1, I make energy quickly. Yes, because it was already stored inside the cell. Number 2, that makes a strong contraction. Absolutely. Because in a strong contraction, we break up ATP fast. That is what glycolysis does. But ultimately, finally, the last point she made is that it's short-lived. I can't do this very long because if I do this too long, I'm going to chop up all of the energy that I stored. So yes, that is 100% correct, Kylie, that glycolysis makes a fast and strong contraction, but it does not last very long. Absolutely. I want to stop talking for a couple of minutes here because you have, it should still be page 11, I believe, right? That you have this picture with the logs in the forest. I need you to summarize what we just talked about on page 11. So how is glycolysis like the logs? How is the electron transport chain like forest? Let's make some summary notes for ourselves. And if you think of questions as you're writing notes, feel free to put those in the chat. In a couple of minutes, I will address those questions. Do me a favor. If you are ready to move on, can you raise your hand for me? I did get a question that came in. Several of us are still working. That's totally fine. And if you can think of more questions, type them in. I've got Alexis's question, so I'll answer that here in a minute. Yeah, so let me address Alexis's question. She asked about basically what the electron transport chain makes. The electron transport chain makes ATP directly. It doesn't have to use glucose. So the electron transport chain is always making ATP. When our goal with glycolysis was also to make ATP. In glycolysis, we had to start with glucose. Glucose had to be there. The electron transport chain completely skips glucose and makes ATP otherwise. So to rephrase your question, what we could say is the electron transport chain continues to make more ATP, and in glycolysis, we run out of glucose, so we can't make more ATP. So maybe kind of a little reframing of that question. Carrie asked about these types of muscle tissues just applying to humans and animals. As far as I know, I think those are the only kinds, somebody correct me who's inside zoology, but I believe just human animals are the only ones who have skeletal muscle tissue. I could be wrong, I'm not a zoologist, but yes, I would say in general, we are just talking about the type of tissues in humans and animals. I think everybody else doesn't have muscle. Any other last minute questions? And I know it takes a while to type, so I'll pause to give us a chance to type if we have any more questions. So I'm going to pull that mean teacher thing that I used to always do in class, right? I'm going to be that mean teacher. Let me switch screens on you and show you. On page 18 of the lesson for today, the end of lesson number 9 on page 18, it talks about 2 kinds of features that are different across my different kinds of muscle tissue. There are different levels of a protein called myoglobin, which you can see shown in brown on that picture there. And there are different levels of mitochondria that I find in different types of muscle fibers. So I'm going to be the mean teacher that's not going to help you answer the questions, which kinds of muscle tissues have a lot of myoglobin, which ones have a lot of mitochondria. But what I will tell you is everything I would have said in class to help you answer these questions, I took the time to type it up for you, and it's written. That, by the way, I get a lot of student questions. They're like, oh my gosh, there's so much writing on these lessons. What you guys didn't realize is how much I talked to you in class. Like, I talk way too much. Full disclosure, I talk way too much. So this page shows you everything I would have said about myoglobin, everything I would have said related to mitochondria. You've got these practice questions here, these areas where you can click your answers and you can check it. These are not graded, so you can use as many opportunities as you'd like. Oh, let me mute everybody. You can go through and answer that as many times as it takes you to get them completely correct. So you have a way to check yourself. But again, I'm going to be that mean teacher, and I'm not going to answer this question for you. So please don't forget about myoglobin and mitochondria. We need to figure out who's got a lot of those things. I know that we did have a question about what kinds of activities we would use these various types of muscle tissues for. So what would be best for fast that fast glycolytic could do? What could let me go to the pictures here to show you. Fast glycolytic versus slow oxidative versus fast oxidative, with some pictures of Olympic sports. What I will say for you with this particular activity, so this activity is on page 20 of the lesson, matching up the types of tissue that would be most helpful for these activities. If you watch the video here on page 19, he goes through and talks about some of the kinds of activities that you would do with each of the types of skeletal muscle tissue. I believe he actually gives the example of at least 2 or 3 of those specific activities. My tip off for you as you're trying to answer the questions on page 20. So here's that video for you on page 19, along with a list of questions for you to answer. Let me show you though. Your tip off here on page 20, when we're trying to figure out which types of muscle tissues would help most with each of these activities, look at the descriptions. So for example, my top type, my first activity that we have listed here is what you would call a deadlift. So a deadlift is when someone literally walks up to something that's super heavy. You can see he's holding a super heavy weight there. You walk up to something super heavy, all you got to do is pick it up and lift it over your head for like a couple of seconds, and then you get to drop it. Now, I say that's all you have to do, right? But it's like, would be terrifyingly hard to do that. All that to say, though, this is something that you have to do for a very short period of time, like you see in the description, but it has to be a very strong contraction. Notice below it when we're talking about tennis, in tennis, you are repeatedly doing a muscle activity. If I do something repeatedly, here's a wink, wink, nudge, nudge, that I'm going to need energy to do that every single time. If I'm doing something repeatedly, I better have a lot of ATP available. And in tennis, we're trying to hit that ball really far, back over the net, hopefully really fast, so our opponent can't hit it back. We need a strong contraction, and we need to do that repeatedly. over a long period of time. The last activity that I asked you guys about is a marathon. When you do a marathon, as they always say, right, slow and steady wins the race. So we don't have to have a strong contraction, but we do need to be able to do it for a long period of time. So when you're trying to figure out which kind of muscle tissue does which contractions, consider those descriptions that are underneath it. If I need energy for a long period of time, for a short period of time, do I need a strong contraction or a weak contraction? Again, I'm going to be that mean teacher. I'm probably not with you, but we can chat it out if we want to have a chat about it in the chat. Let me, here, I'll pull up our pictures. Bear with me. My friends who came to office hours yesterday saw my little Dory thing, picture of Dory that said, so how do you manage all this technology? Just keep swimming. Okay, so here's my 3 activities. And if we are, oops, make sure you got that little microphone crossed off for me. Make sure we stay muted. If we are talking about a contraction that needs to be strong, yes, we're going to have to use a fast ATPase for that. Remember, the faster you chop up your ATP, the stronger your contraction is. So when we talk about the period of time that we have traction for, that's when we have to start thinking about how we made our ATP. So do we have ATP forever, or do we have a limited supply of ATP? That's what it's going to come down to with how long our contraction is for. So here we can help each other out. We said that for my deadlift, that's my picture in the bottom left corner, we need a strong contraction, but only for a period of time. So let's start with that first part. When we say that we have a strong contraction, which kind of ATPase did you guys tell me we need for a strong contraction? Yes, a fast one, right? We got to have a fast one. Okay, so a strong contraction equals fast. We said with tennis too that we've got to be strong as well, right? So tennis is also strong contractions because we got to hit that ball over the net. So we need to have a fast. When we run a marathon, it doesn't have to be a strong contraction. So I hesitate to call it weak, but I'm going to call it weak. A weak contraction, or at least not a crazy strong one, that would be, yep, like P led for us, that would be a slow. Okay, so the strength of a muscle contraction, that tells me if we're fast or slow. And then we're talking about how long we have to do something for. So basically, how quickly are you going to run out of ATP? When I am doing something for a long time, I need to have ATP forever. What I would use for, or what could I use to make ATP forever? What's the word for you can build a lot of ATP? I asked my question weird. Yeah, the electron transport chain helps me to do that, right? And so when I talk about the electron transport chain, if you are a muscle fiber and you use the electron transport chain, I call you which word? Are you oxidative or glycolytic if you use the electron transport chain? Yep, there we go. Oxidative. Absolutely. Okay, so I've got, when I'm talking about doing a contraction for a long period of time, or when I need a lot of ATP, like I need to run a marathon, I need to have used the electron transport chain to make my energy. Hey, when I'm doing repeated contractions, which is a fancy word again for saying I'm going to be doing this for a long time, I am also going to want to use oxidative, still oxidative, because I still need my ATP to last a long time. That's what oxidative means. It's going to last a long time. Over here with my deadlift friend, this is a very short contraction. For a short contraction, I don't need an unlimited supply of ATP. So what's the word I would use to describe a limited supply of ATP? What word would it be? Oxidative or glycolytic for a short contraction? Yeah, glycolytic. Absolutely. Glycolytic. Okay, so look what we've done together. We've used the description of these contractions to help us figure out their name. Anytime we're talking about a contraction that needs to be strong, or we have a really quick contraction, the first part of the name of that muscle tissue that would help us do fast. Anytime we are only doing something for a very short period of time, I can use glycolysis, or I can break down my stored energy to make that happen. But remember, glycolysis, there's a limited supply of this available. So anytime I need to do something repeatedly, like with tennis, or anytime I need to do something for literally hours for a long time, I would need to use the electron transport chain to be making a lot of ATP for me for a long time. So we hashed out using those descriptions if it's fast or slow, if it's glycolytic or oxidative. That's the kind of thing that I would ask you to do or to apply when we are taking our third exam. Help me out before we move on to talking a little bit about acetylcholinesterase. What questions do we still have about powering muscle contraction? And I'll give us some time because it takes time to type. I wish that there was a little, I guess we could do this with emojis. If you all look over in the chat, let me check and see. It looks like they have thumbs up, thumbs down. If you scroll it, give me a thumbs up or a thumbs down. How are we feeling right now? It'd be so much easier if we were in class, right? You could just hold it up for me. Yes, Irini asked the electron transport chain. Basically, it's making unlimited ATP. That is correct. Oh, Kylie died. I'm so sorry, Kylie. Fun fact, I got an email from the school yesterday that said they were closing campus indefinitely. And so I needed to go grab anything that I needed for the foreseeable future. So I was like, well, I don't know how long that's going to be. So I went and grabbed a skeleton. So I got one of those tabletop skeletons that I'll pull out in our lab office hours tomorrow. So my daughter's super excited to have that skeleton in the house and also to have the muscle man. She really likes skeletons with muscles. So that is my 3-year-old. She was really excited about muscle man. So we'll pull those out tomorrow. Yeah, I don't know, Kylie, on the book rentals. Questions that I don't have an answer to right now. So sorry. Okay, well, it looks like most of us are thumbs up or dead. So hopefully mostly up, thumbs up, not dead. I missed all the things that were said. Well, Pauline, maybe watch the video to catch those things. I didn't disinfect the skeleton, but the thing is, no one's been in the lab since before spring break. So I think that I should be okay. I'm hoping that I should be okay. We'll know in about 14 days, right? I think it should be good. There's been no one up at school with me, so fingers crossed, I guess. Yeah, Pauline, I will get the recording of this posted tonight, so go back and watch that. Feel free. That'll be available for you. Okay, let me go back. The other thing that we'll talk about, and again, I'm gonna pull that mean teacher thing. I am not going to talk through sarin and Botox with you entirely because it's really important. Those are two really important topics to me that I want you guys to hash out. They are critical thinking topics, and I know that's where you come with questions, but I'm not going to give you the answers to those questions right now. I want you to try to work it out, especially since half of us haven't really had a chance to talk. I'm just going to be honest with you, full disclosure right now. I'm not going to talk about sarin gas and Botox specifically, or at least not give you the answer. The best thing to do, Carrie, is take notes on what we talk about now. Come back on Friday with proposed answers to those questions, and we can chat it out together on Friday during Friday's office hours. What I will talk to you about today is remind you of the importance of acetylcholine, which we talked a lot about in our office hours yesterday, and I'll talk to you about why acetylcholinesterase is important as well. So the first thing I want to ask, because it was a big topic yesterday, who can help me out with this question here? What is the purpose of acetylcholine, or what does acetylcholine do when we're talking about, well, so not acetylcholinesterase. I actually want to specifically talk about acetylcholine. I'll circle it here on my picture. Who remembers yesterday when we were talking about acetylcholine? What was the job of ACH, acetylcholine? Yeah, it's a message. Absolutely. This is the message that I use to send from a neuron to a muscle cell, telling it to contract. So a muscle cell is constantly listening and constantly waiting for a neuron to send it the message to contract. If it sends it the message to contract, then we go through the process that we talked about yesterday. I'll type up its big long name, right? Excitation, contraction, coupling. We said yesterday that excitation meant that a neuron needs to send a message to a muscle cell. If that neuron sends the message to the muscle cell, which is the acetylcholine, it's going to lead to the contraction part of its name where the muscle cell contracts. So excitation, contraction, coupling starts with a neuron spitting out acetylcholine. And this picture, by the way, I'm going to reference back and I'll tell you, the picture we're looking at right now comes from page 12 of your lesson for the week. Slightly modified version of what you have in your printed lecture notes. You may want to go back as you're working through the end of lesson number 9. You may want to go back and write down the things that are over here. These things that are over here, you'll find them on, let me type it instead of write it, page 12 of lesson number 9. I modified the picture just a little bit to show you what's going on. So let's talk about what's going on in our picture here. First thing for us to know in our picture here is my top part of this picture is showing me a motor neuron, or it's showing me a neuron whose job is to talk to muscle tissue, to tell the muscle tissue to contract. So here's my neuron with its message. The message that is inside of it is this little thing you see right here, ACH, acetylcholine. And I mentioned this yesterday, I'll say it again, that when we talk about acetylcholine, ACH on the exams and on the homework, I will give you both the full word and the letters ACH. So don't worry about memorizing how to spell acetylcholine. I won't make you do that. But I will give you acetylcholine in parentheses ACH. Okay, so motor neuron has acetylcholine. The motor neuron is wanting to make its muscle cell that it talks to contract. So first thing that happens, it spits out this acetylcholine that was inside the neuron, spits it out, and it goes down and it attaches to, here's where we start talking about yesterday's lecture, it attaches to chemically gated sodium channels. Here's a review question, and I would ask you this question if we were in class together too. When I talk about a chemically gated channel, it is or is not always open. A chemically gated channel. Correct. It is not always open. That's what that word gated means, right? Every time gated means we're not always open. This is a chemically gated channel. What do I use to open up chemically gated channels? What opens up that kind? Chemically gated. Chemicals. Yep, absolutely. Acetylcholine is a type of chemical. So, step 1, my neuron spits out a chemical called acetylcholine. Step 2, that chemical comes down here and attaches to a chemically gated channel, which allows sodium to rush inside my muscle cell. Now, we talked a lot yesterday in office hours about what happens once that sodium goes inside and how I move my action potential places. I'm not going to go back through all of that, but I would really encourage you to look at that recording from yesterday. The big idea is that once I rush in all of this sodium that we're talking about, my chemical being able to open up, once I rush all that sodium inside, that will trigger a muscle contraction. It's gonna send that message across the membrane of the cell. It's gonna go all the way down and make that cell spit out calcium, which ultimately leads to muscle contraction. So that's yesterday's video. That's not today's video. But here's the thing. Once I have brought in a bunch of sodium inside my muscle cell and it has depolarized, or it's gotten the message that it should be contracting, at that point, I'm done with acetylcholine. I don't need acetylcholine. So when we look at it down on the receptor, once that receptor has opened up, once that channel opened, it brought in sodium and the muscle fiber has depolarized, now we're going to spit out the acetylcholine. It stops being attached so that this channel can close. Except I don't want this acetylcholine neurotransmitter to just float around forever next to my muscle cell. If it floated around forever next to my muscle cell, it might go find another chemically gated channel to open up and make the muscle cell contract again. So when I've contracted once, because that's the job of acetylcholine, to make me contract. Once I've contracted that one time, once I am done with acetylcholine, it will detach from the receptor and it's going to bump into this guy right here. So at the bottom of the image labeled number 3, you see a thing called acetylcholinesterase. Acetylcholinesterase. And we can be glad that we don't have to spell that one on the lab exam, right? So acetylcholinesterase, I will abbreviate that ACHE. The job of acetylcholinesterase is to break down acetylcholine, like its name talks about. So yes, it breaks down the acetylcholine. So we're done receiving the acetylcholine message. My muscle cell has depolarized. My muscle has contracted. That acetylcholine releases my chemically gated channel, and it floats over and bumps into this enzyme called acetylcholinesterase. Acetylcholinesterase does a little ninja chop and breaks it in half. It goes from being acetylcholine into a little piece called acetate and a little piece called choline. The reason I want to chop it up is because if choline by itself bumps into my chemically gated sodium channel, the channel stays shut. It's not going to open just for choline. And the same thing with acetate. If acetate bumps into my chemically gated sodium channel, that channel's still not going to open up. It's not going to send the message. So when I'm done hearing this message, once it's depolarized the membrane, the message floats over, bumps into its friend over an enzyme called acetylcholinesterase. That's going to chop it up and make sure that I don't keep hearing that same message. That message is gone. So acetate will float away. I don't need it. Sometimes my neuron will take back this choline part because, like, hey, it worked really hard to build acetylcholine. It's gonna take choline back inside and it'll recycle it. But choline by itself and acetate by itself cannot trigger muscle contraction. It's just acetylcholine when it's together in that whole piece that's going to allow us to have muscle contraction. So acetate and choline together open the gate. When acetate and choline are together, what I call them is acetylcholine. So those two pieces by themselves wouldn't open the gate. Only when they're attached to each other chemically, acetylcholine, that would open the gate. Correct. What other questions do we have so far? You can't believe you understand that. Hey, that's great. I'm glad to hear that's what you can't believe, right? That you understand it. That's awesome. While questions might still, okay, here's another one. The role of acetylcholine is to open the gate. Yes, that is the job of acetylcholine, is to go down and open up the chemically gated sodium channel to tell my muscle cell to contract. So here, I'm going to pull up a whiteboard. I have two questions that I'm going to pose for you that I want you to write your answers to, and then we're going to talk them out in the chat. First question, what would happen at the, I'm going to use a word from yesterday, neuromuscular junction? That's the place that we just looked at on the picture where a neuron meets a muscle cell. Okay, so what would happen at the neuromuscular junction if, here's our two questions. First question, acetylcholine never got broken down by acetylcholinesterase. That's our first question. We want to predict based on what acetylcholinesterase does, what might happen if it doesn't break down acetylcholine. The second question is, so what would happen at the neuromuscular junction if the motor neuron never released any acetylcholine? Here's our two questions, and I want you to write down some notes for yourself. We want to predict what might happen if acetylcholine never got broken down. Carrie says she can't see anything. Do I have anyone else that can't see anything? Me too. I'm trying to share my whiteboard. Odd that some people can't see it. It's not a picture. It's just a whiteboard with two questions written on it. So we're like 50-50 split. Okay. Well, for cannot see, here, write down for me. Yeah, you could try logging in again, or I'm not sure what popped up. Here's the questions. So you can write down these questions in your notes so you can answer them. The first question, what if acetylcholine never got broken down? That's the first question. Okay, someone's saying we're fresh, and that works. That's awesome. Okay, so what if, I'll type them into our chat too to make sure we get them out there. What if acetylcholine never got broken down? That's the first question. And what if acetylcholine never got released? There's our second question. So if the refresh doesn't work, there's our questions to see if that helps. Perfect. Sounds like the refresh is working. Good. So I'm going to give you a couple of minutes to write down your notes, your predictions of what we think might happen in both of these scenarios. What might happen in both of these these cases here. To help us with these questions, I'm actually going to pull up a picture that we spent time on yesterday. This picture shows us the entire process of excitation, contraction, coupling, going from a neuron spitting out a message to that message causing my muscle fiber to freak out and go positive, to the entire membrane freaking out and going here in the middle with my T tubule taking the message down, all the way down to the process of muscle contraction. For my friends who couldn't make it to office hours yesterday, this is kind of the picture that you want. You know how in class I used to tell you guys, go stand in front of a mirror and talk the process out? Here's the picture that shows you everything from beginning to end. If you can explain how muscle contraction works using these 3 images, you're good. You understand the process. If there are certain parts that are hanging you up, that's where we want to chat with classmates. That's where we want to send me a specific email, parts of it. But here's kind of your summary of how the process of excitation, contraction, coupling works. So what we're talking about right now are 2 possibilities of problems I could have all the way up here at the beginning. So I zoomed in on the first part here. This is kind of the part yesterday we called the excitation part. So when a neuron talks to a muscle cell, that's the part that we're focusing on right here. And I ask you 2 questions. I asked you, number 1, although I guess I made it number 2. Number 2, what would happen if a neuron does not spit out acetylcholine? And number 2, what would happen if the acetylcholine that got spit out kind of stuck around forever? So let's start with our scenario where no acetylcholine is released. Let's start with this one here. Light me up in the chat again, because my what are some of the things that we said would happen if my neuron did not release any acetylcholine? What might happen? Okay, the change in the membrane charge would never happen. I love it. Yep. So no membrane charge change. What else? I know we said a lot of different things. The first thing that's going to happen, no membrane charge change. Yeah. And Carrie just went for the gold, right? That's the ultimately, if I don't change my membrane charge, I have no contraction. No contraction. Acetylcholine must be released. When acetylcholine is released, it goes down and causes a change in the charge of my muscle fiber. The change in the charge of my muscle fiber is going to allow it to contract. So if my muscle fiber, if my muscle cell never gets the message to contract, it will not contract. Yeah, it'd be like no message was ever received. Yep, because acetylcholine is my message. So first correlation we could make at the neuromuscular junction. If there is no acetylcholine that's being released, my muscle will not contract. Let's talk our other option here. So we're going to go kind of to the opposite end of the spectrum. I won't make you type acetylcholinesterase because that's a big, long name. But help me out. What did we say the job of acetylcholinesterase was? That should be easier to type. What does acetylcholinesterase do? What is its job? Well, so we're getting a little mixed up between acetylcholine and acetylcholinesterase. Acetylcholine is the message. That's what we just talked about. Yeah, a couple of us have picked up on it. Yes. Acetylcholinesterase, I'll write its whole name and then I'll abbreviate. Acetylcholinesterase, or the easy way, ACHE, that's its abbreviation. Its job is to break down acetylcholine. Acetylcholinesterase breaks down acetylcholine. We need to break down acetylcholine to prevent the other thing I asked you to make a prediction about. So the other thing I asked you to make a prediction about is if ACH stays forever. That's what we'll say. If it stays forever, if acetylcholine does not break down after I've received the message once, what are some of the things you guys predicted would happen if I didn't chop it up into acetate and to choline? What are some things that might happen? Yeah, I like it. Leslie says it would find another channel to open up. Absolutely. The muscle would continue to contract, or we would see what are called muscle spasms. Absolutely. So if the acetylcholine stays around forever, maybe the simplest way to summarize it is it leads to constant muscle contraction. Let me enter that. I want to make sure that muscle contraction. Because acetylcholine would open up, let me pick another color. First, the acetylcholine would open up this channel right here that I circled in black. And then once it had depolarized that membrane, it would bounce over and then it would go open up this channel. And then it would bounce over and go open up this channel. And it's just going to bounce around and keep opening up channels and opening up channels and do this forever, leading to my muscle constantly contracting. Or yeah, I think someone used the terminology kind of like a cramp or a spasm. When we send the muscle the message to contract, we're typically not sending a message to contract like forever. So if I keep sending this, if my muscle keeps hearing the same acetylcholine message that was sent 5 minutes ago, my neuron wasn't trying to tell it anymore that it's time to contract, but it's going to keep contracting. So we need acetylcholinesterase to get rid of muscle contraction messages once I've heard them. We need to release muscle contraction messages to begin with to make sure that my muscle can contract. So here's what I'll tell you and what I'll give you for your readings, and you're working on how sarin gas works and how Botox works. With these two disorders, we've got a problem with acetylcholine. The kinds of questions that you would want to ask for yourself, here's in the most basic way. Ultimately, where I want you to get when you're done studying how sarin gas works and how Botox works, and don't put your answer in the chat, by the way. Don't do it, because I don't want it out there. This is where I want you to get to. When you read about sarin gas and you watch the video about sarin gas that talks about how sarin works, ultimately, it's either going to boil down to having too much acetylcholine or having too little acetylcholine. At the end of the day, in the most basic terms, that's what sarin is a problem with. Either too much acetylcholine or too little acetylcholine. Same thing is true with Botox. As you are studying how Botox works, it's either going to come down to there's too much acetylcholine, there's too little acetylcholine. I want you to understand how these processes work. You don't have to know super intense chemistry on how they work, but you do need to be able to reduce it down to, is there too much acetylcholine? Is there too little acetylcholine? We made some predictions about what would happen if I had too much or too little acetylcholine. So use that to help you out as you're trying to figure out what's going on with sarin gas versus what's going on with Botox. Okay, so I'm going to pull up my meme, And I am going to open the floor here. We got about 10 minutes left. I told my friends yesterday, by the way, that I made this for you guys. Like, we got to have some humor, right? So I made you this meme about Botox. You'll learn more about it if you haven't already done that part of this week's lesson. You'll get to learn a little bit more. Botulinum toxin, which is what Botox is, it's like literally the most dangerous thing known to man. And we put it in our face. Like that's the state of our vanity, right? We put these toxins in our face. So I know it's real crazy. So I'm going to open up the floor to questions that we have, either about the stuff we talked about today or lab stuff. If you have procedural questions, I'm not going to answer content questions for lab yet. That's tomorrow. Or any kind of general questions we have here in the last 10 minutes here of office hours. What questions do we have now that I can help out with? And here I'll try my attempt again today. Telling you, by the end of online class, I'm going to be real great at drawing you guys penguins. We're going to get there. Life goal. It's already looking better today, man. I'm impressed with myself here. Look at that skill. How can we look at stuff we missed on the first exam? That is an excellent question. I did not think to grab your exams before campus locked down. So honestly, I don't have a way for you to look at the first couple of exams, and I'm very sorry about that. I did not think of that. What I will tell you is the final exam is cumulative, but it is not everything cumulative. Let me add to my list of what to work on. I will try to get the final exam objectives list to you as soon as I can. Yes, as we get closer to the final exam, I will definitely schedule some office hours where we can come in and talk about specific learning objectives that you have questions on. So yes, I'll can to help us get as much support as we can without those exams, since they are locked in my office. Deepa, the course calendar has changed. Look at the updated course calendar. They will not be on the same day anymore. For my Friday lab, they used to be. They are not anymore because the schedule has changed. So look at the updated course calendar to see the new general schedule. Can we have the extra handout on excitation, contraction, coupling? Can the extra handout be used for the page 8 exercise? You can use whatever resources are helpful. So check out the supplemental resources folder back from our normal lecture things. There are some handouts in there that go along with those videos. Please feel free to use anything that is helpful for you. So definitely use that handout if it's helpful. I do still have grading stuff like Quiz 11, like Anatomy for Babies, that I will get to eventually. I am building guided lessons right now, so that my head is down and I'm working on those things. Grading will happen as it can, but it will happen as soon as I can get to it. But right now I'm really focusing on getting you guys the resources that you need. Update about when the schedule changed. Not since the online transition. It's just changed as of what we started at the beginning of the semester. So if you went in, it was probably Tuesday or Wednesday of your second spring break week. That's when I posted that calendar. That's what it's updated as of. So not a new one. Penguin goals, right? My penguin is looking pretty good. Oh, it's not ears, Leslie. These are, I'm a fan of macaroni penguins. So this is macaroni penguins. Those are little, here, I'll turn his feathers yellow. Those are the little yellow feathers that come off of macaroni penguins up there. So we'll make today's penguin here. I'll make today's penguin a little girl penguin. We'll pretend it's my daughter here. who's done a very good job not coming in today. I am impressed with her. Alexis asked about extra credit, still coming up with something. So yes, there will be, yes, thank you, Grandma, right? There will be something extra credit. I don't know what at this point, but I know that Anatomy for Babies was our primary way to do that. So still in process, I will come up with something for you to give you that opportunity too. Pilar and Nora, you're welcome. I'm happy to be able to help you guys out as much as I can. Yeah, definitely a curveball for me as well, going online in the middle of the semester. I think I told some of you guys that I'm planning to teach online in the summer. That was always the plan. So you guys are at least lucky that I was thinking ahead about how to do an online class, because a lot of your teachers are like, never in my life would I ever teach an online class. And all of a sudden they didn't have choice. So at least you guys had an instructor this semester that was thinking about how to build an online class. You at least have that benefit going for you. So yeah, you are my guinea pigs. Hopefully things work together. Online group work, it essentially is replacing Anatomy for Babies in the sense of that is where those points that some of those points that would have been Anatomy for Babies are that's why we're doing the group work, to get the same kind of assessment. Technically, the Anatomy for Babies category is closed. Those online group works are going into the homework assignments category. But yes, it's essentially replacing Anatomy for Babies. But I probably won't do extra credit in the group work. Yeah, Eileen mentioned she's got a psych instructor that isn't quite as involved. Just be patient with them. Give them grace. Gonna be honest, it's real tough. I'm happy to be involved and to help you guys and support you guys, but it's real tough. So especially those that are stretched even thinner than I am, I could see it being really, really tough for them. Yeah, so the things that are due this weekend. Yeah, this is a good review and a good thing to pass along to your friends and group me. Sunday night at 11:59. So mentally make it 11:45 to make sure you're done. We need to have lesson number 9, homework assignment for lecture, done. That's all this muscle contraction stuff. We also need to have week number 11 lab assignment done by 11:59 on Sunday. So 2 things are due, lesson number 9, as well as that lab activity. Yes, for my Friday friends, not due Sunday. Anatomy for Babies is due Friday morning at 9:30. So that would be a good thing to remind your friends of and group me. Anatomy for Babies needs to be submitted to me in that Dropbox by 9:30 on Friday, because that's when you would have given it to me if we were still in person. That's when it would have come in. So that's coming in Friday for my Friday lab friends. What other questions do we have? Yeah, start playing with it now. Eileen, she was asking about how to submit those Anatomy for Baby cards. Start playing with it now. Take your pictures, try to get them into a Word document, and then save that Word document. And so start playing with it today, because it's Wednesday. Needs to be up by before Friday office hours start. So please start playing with it now to get it to work out for us. Well, my questions aren't coming in as rapidly, so here's what I'll say in closing. Again, tomorrow we're going to focus primarily on lab. So as much as you can, please try to work through, here I'll pull it up and show you guys. Please try to start working on your guided lab activity. So the guided lab activity, remember your lab sheet for this week that you printed before break is still totally valid, or even when you go in and print it now, your sheet that you print is going to say week 10. You don't care about the week number that's on the sheet. You care about the fact that it says nervous tissue and muscles and bones of the leg. This activity walks through one by one. So first thing you're supposed to do in that activity was we were going to pull out a microscope and look at nervous tissue. Obviously, we're not in the lab, so we're not going to pull out a microscope, but if you have a student Google Drive, you can get those images of nervous tissue. We want to make sure we can label the pertinent structures. Here's some pictures to help you out with identifying things on nervous tissue itself. That's exercise number 1. And then in exercise number 2, we were looking at our 2 neuron models. So here's a video that completely overviews the structures of the neuron models on our exact model. We're going to make you do a little bit of critical thinking, but also you've got that labeled images PowerPoint that'll help you label the other model. So this goes through one by one, exercise by exercise, to help you out with the labeling, to help you out with the functions. Use this guided learning activity. Please try to get this done before class tomorrow so you can come to me with specific questions that you have about the activity. Let me see what questions we got popping up in here. This is our last time going over lesson number 9. On Friday, I will also take specific questions that you guys have over lesson number 9. This is the last structured time, I guess you could say, that we're going over lesson number 9, where we said we were specifically talking about the second half. But if you still have questions, bring them Friday. Update the lecture stuff online, paper documents. So I presume, Carrie, you were talking about updated lecture number 10. Yes, I was up late last night. It is almost done. You will have it by end of day today. It might be end of day. It might be midnight, but it's going to be up before I go to bed tonight. So not up yet because I'm finishing up that lesson and finishing up the update. So hang tight with me. That will be coming soon for you. Technically, with lesson number 10, that assignment's not going to be due till next Sunday anyway. You're going to have lots of time. Understand wanting to make sure to be staying on track. So by tonight, before I go to bed, you will see lesson number 10 and the updated lesson number 10 outline posted. For my Friday lab friends, question about anatomy for babies, you are not emailing it to me. You are putting it in the Dropbox. So check out in your course in the anatomy for babies area of Blackboard. If you click on that, the very last thing in that area is a Dropbox for that submission. So go over to the anatomy for babies folder. You will find a Dropbox in there. Letty says, thank you for the videos. Glad the videos are helping out. And yes, Pauline and all of you, happy to go. You too, Leslie. Have a great day. All of you guys have a great day. I will be here tomorrow, same time, same place. So hope to see many of you. And again, pass along in GroupMe anything that you feel like would be good for them to know in a time-sensitive matter. Go ahead and pass that on to them. Yeah, everybody stay safe. Great to see you guys all this morning, and we'll just keep trucking along. So yes, everybody stay safe and have a great day. You're welcome, Irini and Rosa. Yes, glad to be able to help. Correct. Carrie asked if this is going to be the schedule for the rest of the semester. That is correct. I have not heard from students that this timing is terrible for everyone. And this was when I put out that poll for voting. This was the time that people said they wanted. So I'm going to plan Monday through Friday, 9:30 to be here for the rest of the semester. So that is the plan. I think I'm going to go ahead and go off camera. I'll leave my mic on here. But yeah, feel free to keep sending in questions that you have. I'll leave the room open for a little while longer. I will go ahead and stop our recording though, since I think we're done with most of our content.